Welcome, everybody. It's my pleasure to introduce Leah Johnson. Leah is a physical oceanographer at the Applied Physics Lab at University of Washington, although she spends much of her time down here. She received her MS from San Francisco State, and I didn't know this when I invited her, but she does have a Moss Landing connection. She took chemical oceanography from Kenneth Cole and took the scientific diving class, so has something in common with many of you in the audience. Got her PhD at the University of Washington, did a postdoc at Brown University, and is now back as a senior oceanographer and affiliate assistant professor at Applied Physics Lab and the School of Oceanography at University of Washington. So her research combines observations, numerical models, geophysical fluid dynamics, applied math across scales to address air-sea interaction and distribution of physical and biological tracers. So we'll hear a lot more about that in relation to the North Atlantic spring bloom. So please welcome Leah.
So Tom pretty much just stole my introduction slide, but I will just add to that. Oh, by the way, I'm really happy to be back here. Like Tom said, I have some fond memories and remember learning a lot from this campus. And plus, it's beautiful to come visit. So I did my my master's in marine chemistry, but I really am a physicist at heart and did my PhD in physical oceanography. And I think a lot about the upper ocean, so both boundary layer turbulence, surface force boundary layer turbulence, as well as submesoscale and mesoscale processes. And so it's in that kind of framework and knowledge base that got me interested in biophysical interaction, and ultimately led me to the North Atlantic exports program in the North Atlantic. But I'm humbled through my experience. I'm humbled by how much broad expertise is needed in order to address a problem like quantifying carbon export out of the ocean. And so this is just a small subset of the team, but people that I have been working closely with and who have contributed to the work that I'll be showing today. So just want to acknowledge that. Right. And also, this is like a physics-centric part. This is viewed through an ocean physicist. All right, so we've all seen pictures of this. We all know that NASA's satellites orbit the Earth, taking beautiful images and collecting important information about the Earth's surface, such as this one. This is the North Pacific. Here, let's see if I can figure out how to use this pointer. Yes. We have Alaska. This is the North Pacific. This is the Inside Passage. And of course, in the ocean, what we're looking at is evidence of massive amounts of phytoplankton. And the role of these marine primary producers on the fate of organic carbon in the world's oceans is a major goal of NASA's carbon cycling initiatives, and was really the backbone of the NASA Exports Field Program, specifically aimed at quantifying the different pathways of carbon export out of the upper ocean. What I always love about images such as this are the swirling patterns that you see in the ocean, and we're reminded that plankton also means floater of the sea, and these primary producers are being carried around by the ocean currents, highlighting the submesoscale and mesoscale turbulence that defines the upper ocean. All right, so more about the export's goals. You can divide them up into sub-goals, which includes determining the role of upper ocean ecosystems in the vertical transfer of matter when quantifying these organic carbon pathways, and the processes that control the efficiency of this carbon transfer away from the well-lit surface ocean. And then also, how do we use insights gained from these field programs under this framework to reduce uncertainty in carbon export estimates. And so lots of the work to be able to answer these questions can be thought of in a conceptual framework, such as these boxes and arrows on the right-hand side that describe these carbon export pathways. But of course, the ocean isn't just one big box model, and there's different arrows in different boxes that can be used to describe carbon export pathways in different regions of the world's oceans. So it's important to study carbon export in more than one place. All right, and so thus, exports field programs focused on two very different regions. And so on the left-hand side, we're seeing the field site of the North Pacific Exports Field Campaign, and this is a region near Ocean Station Papa where upper ocean dynamics are thought to be pretty one-dimensional surface forced, and in this case, the boxes and arrows in this diagram are thought to be dominated by biological processes. To contrast against this environment, the second field program, which is the field program that I was a part of and that I'll be talking about today, occurred in the North Atlantic. And so this is a very eddy-rich region where you have water masses crashing into each other and being stirred by an energetic eddy field. In this area, the pathways physics is playing an increasingly more important role on top of all of the complicated biology that's happening. All right. So again, I'll be talking about the field program in the North Atlantic. And so one thing that so this region, the point was to study biological processes in a very interesting place. Well, it turns out that the fact that the place is really interesting also makes it really hard to observe it. And so an immense amount of effort and planning and execution, design went into executing this field program, including weeks before or months before the program the ships ever set sail, we wanted to, so the point was to identify a mesoscale eddy in the North Atlantic that was retentive and focus our efforts on the center of that eddy in order to conduct biological rates experiments, while simultaneously sampling around the environment around the eddy and around the eddy edge in order to observe the interesting lateral variability. And so months before we ever set sail, we used geostrophic fields from altimetry, velocity fields, in order to identify eddies and track them. We met weekly and looked at how the eddies moved and tracked over time. And when we decided on an eddy, we finally sent out sentinel sea gliders. And the sea gliders checked these different eddies to make sure that they really did exist, not just in satellite data. And from there, we felt confident about this one eddy, which is this orange dot here. This eddy was intensely sampled, and through that we were able to create kind of paint an environment, a physical environment of what the eddy looked like and how it evolved during the month-long field program. And from that physical environment, many other biological and ecological and process studies can be painted within that. And so I'll be talking a little bit about that physical environment. It's published in we call it the context paper, but it's got a much longer name, but it's published in Progress in Oceanography. And then I'll be talking about some examples of how we can quantify carbon export within that environment. And this is mostly work in progress, thinking about looking at particle sinking inside the retentive eddy, as well as lateral stirring at the surface, and finally, evidence of vertical advection around the eddy periphery. Right. So I think I have to move this from here. So like I said, this was we had 3 different ships and a bunch of autonomous platforms sampling. And in the corner, you can see this little eddy center. So we were tracking the eddy center with satellite fields, and all the sampling occurred in the eddy center reference frame. We had a Lagrangian float, a sea glider, and a BGC Argo float that tracked the eddy center. So we have both satellite estimates and the assets that agreed, and a process ship that followed the eddy center and did process studies. And then we had a survey ship and a bunch of surveying gliders surveying around, and you can see evidence of us running around. You can also see some of us running away all at the same time, and that's a result of some pretty vigorous storm events that happened, which end up becoming a part of this story. All right, and then we pick up our assets and we go home. Okay. So this is in the eddy center reference frame. You can see the satellite geostrophic fields and the glider sampling both inside the eddy center and around it, and really characterizing the chlorophyll around this eddy. And so this is an example of a way that we can take images of chlorophyll derived from satellite images and really continue to map them down further into the ocean through these biooptical measurements. But of course, chlorophyll is only one part of this story, and the rest really had to be done with hardworking scientists on board ships, and in this case, hardworking scientists on board 3 different vessels. So the process ship was the RSS Discovery, which Colleen was on, and that did rates experiments in this eddy center. We have the survey ship, which Sasha and I were on, that sampled all around the eddy and eddy periphery. And we were joined by Woods Hole's Ocean Twilight Zone experiment, which also did many process study and survey experiments, which we'll be talking about. All right, so what does it mean to have an anticyclonic mode water eddy? This actually ended up being a really cool place to do an experiment. We didn't know it at the time until we sent our sea glider in, but this eddy has a really interesting signature. Hopefully you can make it out from this movie. On the right-hand side, we're looking at depth and distance across from the eddy center. And these are isopycnals, both temperature and salinity. And what you're seeing here are these sloped isopycnals that show you the anticyclonic nature of the eddy, which is what you see from satellite. But actually in the surface ocean, where all the action is, the isopycnals are going the other way, creating this deep mode water. So this is that low PV, low stratified core, which is outlined here in gray. And this is what helped keep the eddy, in physical oceanography terms, this is a low PV eddy that helped keep the eddy retentive and ended up being a very good place to study these dynamics. A little bit of something that we also didn't expect is that this diamond structure made it really easy to know when we were in the center of or right above the center of the eddy. We call these surface core waters. And so whenever you're at this widest part of the diamond, you know that you're kind of in the place where you need to be thinking about the eddy center dynamics. And for anyone who's done oceanography, you're just poking holes in the ocean. It's really hard to be able to map things out in a moving framework, and this ended up being a really convenient way to map everything in the eddy center reference frame. Okay. All right, so in addition to this mesoscale eddy that we're seeing, I should be cognizant of time. In addition to this mesoscale eddy that we're seeing, a lot of the action is happening in the near surface ocean, which is the surface forced layer. And so your canonical view of springtime in the North Atlantic is that there's deep winter mixing. All right, we're all good here. The heat flux changes sign, that's this. So this is wind stress, this is heat flux, and this is mixed layer depth. Okay, so mixed layer depth taken from the sea gliders, and then this is when we were out there. So this is the sea gliders before we were out there. So in mid-April, the heat flux changes sign, and whoop, the mixed layer depth shoal, that's like, what you expect to see there at this time. And from here, chlorophyll begins to increase. Great, we're gonna go out and look at what happens in this perfect environment. But then not too long after we get out there, we get pummeled by these different storm events. And it really deepened the mixed layers. It sent us driving away because the wind state got really challenging. And it also changed the upper ocean a little bit. And so we think about a little bit about how these storm events impact the evolution that we see. All right, so I've been kind of saying how cool it is that we've been able to map the eddy center, and this is just more evidence of that. So this is, in the background, we're looking at the sea glider temperature and chlorophyll measurements of the sea glider that was in the eddy center. And through a really rigorous intercalibration program, which you need, especially when there's variability, because you can't, if one ship is measuring one temperature and you're measuring the other temperature, you have to make sure that they're calibrated in order to know if it's a real difference. So we had a very rigorous intercalibration program, and what we're looking at here is any asset that collected a profile in the eddy center is laid on top of it. And so this is a real success of both our eddy tracking efforts and our intercalibration program, that all of these line up. And it means that when you take a CTD cast and you think it's in the eddy center, you can be sure that it actually indeed is in the eddy center. And it's not only for chlorophyll and temperature, but we have this for other bio-optical measurements and nutrients. And what does this tell us? Well, here we're looking at some different variables and how they changed in the eddy center. The eddy center is blue. The eddy center is blue, and the pink and green are different water masses outside of the eddy center. And you could see that if you were to squint your eyes, you would mostly see that in some of these temporal variability is larger than spatial variability, which is an interesting result in and of itself. And if you squint your eyes, you might see that throughout the program, chlorophyll decreased as well as POC that was estimated from backscatter. So there's a long time scale evolution here. Embedded in that, there's some finer scale changes, a result of these storm events. You can really see it clearly in oxygen, where oxygen gets drawn down during the storm events. And then you see it becoming more supersaturated in between events as these communities begin to grow again. And you see that is mirrored in an opposite way by the nutrients, showing that there's both a long time scale evolution and some perturbations from these storm events. Okay, so what can we do with these? This sampling, this time series inside of a retentive mesoscale eddy? It turns out you can do a lot. I'm sure there's many more that can be talked about, but I'll be just talking about a few that I have been a part of, and including these results, which is being written up by Dave Siegel with collaborators like Colleen and Sasha and myself. And we're looking at UVP data, so this is taking pictures of different size classes of particles in the upper ocean, so it's estimated size class on the x-axis, where bigger particles are over here, smaller particles, and depth. And this is only UVP data taken within this eddy center, so we've already identified the profiles that can be used here. Oops, let me go back. Okay, so so what we're seeing here is that in the beginning we have some small particles in the mix layer. The distribution of larger particles begin to grow and then begin to sink out. And this is consistent with other measurements, such as particle traps and gel traps that were taken in the eddy center, seeing this event of sinking particles out of the mix layer. So this is carbon export. This is a time series of carbon export out of the mix layer, which is pretty cool. And with the fact that we have a time series of it, we can actually get rates. I think Dave estimated somewhere around 30 meters per day of these particles sinking out of the mix layer. Right, but you need a consistent rate measurement, and if you didn't, if you included all the UVP samples and weren't able to isolate the ones out of the eddy center, these would be included in your rates, and these were collected kind of early on. These are at the eddy edge, and so these waters have a different time history, which means that just in a matter of 80 kilometers, you can get waters that have a very different time history. And if you weren't able to isolate them from your data set, they would, you wouldn't be able to get this consistent time series. So this is just a really nice result. All right, so that's an example of some really nice work being done in the eddy center. But we also know that there's a lot of lateral stirring. And you can see that in this image of satellite chlorophyll on the left, and satellite SST on the left, and chlorophyll on the right. And what we're seeing here is the cold, fresh polar waters coming from the north, crashing in with the warm, salty waters from the Gulf Stream to the south, and then they get stirred around by the eddies. And so you're seeing that here. You're seeing this warm water kind of being siphoned around into these kind of smaller, what we call, sub-mesoscale structures. And you could see that there's a similar kind of patchiness in chlorophyll. And this implies this patchiness in chlorophyll amplitude also may imply a patchiness in phytoplankton communities. And so that is indeed the case. Sorry, I'm still trying to figure this thing out, which is amazing. Okay, it's really nice too. Okay, but in addition to mesoscale stirring, we have these storm events, and it turns out that really strong wind events are associated with Ekman currents. So on top of the fact that you have these geostrophic velocities kind of swirling particles around and water masses around, you end up having near the surface, you can have wind that pushes water around on top of that. And so we explored the effect of that in terms of the retentiveness of the near surface waters of our eddy by doing particle tracking. And we particle tracked backwards in time particles that were only moved around by the geostrophic fields, and that's our purple particles right here. This, shows what we've agreed what we've hoped all along, that this is a retentive eddy. So if you're advected around by the geostrophic fields, then you're going to stay within the eddy. But if you also on top of that include Ekman currents and then also backtrack it, which is the green lines, you can see that particles can actually come from outside of the eddy. And so that means that some of the surface waters inside of the eddy might have had an origin outside, and that some of that exchange might happen during these events. And right here I'm just showing retention times inside the eddy. So if you're moved around by geostrophic currents, you have a longer time inside of the eddy. But if you are moved around by geostrophic currents and Ekman currents, you have a shorter lifetime inside of the eddy. And so this idea was explored further by a graduate student, John Sensausi at MIT, working with Heidi Sosik. And he's really a mathematician thinking about ways to use machine learning in interesting ways. And he took IFCB data, which we all know can be very large amounts of data. And using non-metric multidimensional scaling, he was able to reduce this into different communities. And then he used KL divergence statistics in order to say how different were different communities from each other. And with those results, he was able to identify that before the first storm, there was a community that made up a certain region of parameter space inside of the eddy, and a different community at the warm filament outside of the eddy. But then after the first storm event, if you just go inside of the eddy, you can see that the community structure is a mixture of these two. You could see similar patterns in the temperature and salinity mixing profile. You could see this in temperature and salinity space, suggesting that, in fact, after the storm event, the eddy center now contains a combination of different communities from both inside and outside the eddy. So that was a really nice result. And that's submitted. All right, so I've talked about carbon export in the eddy center. And we've talked a little bit about lateral stirring and Ekman dynamics and how that might impact phytoplankton communities inside and outside of the eddy. But another thing that the lateral stirring does is it creates very interesting physical dynamics around the eddy edge, particularly these strong density gradients, which we call fronts. And those fronts are associated with strong circulations that not only move things around laterally, but can move things down vertically, which offer an alternative way for organic matter to get down to the depth, different from what we were seeing before with the sinking rates. And so we knew we were interested in studying this problem. It was a very difficult problem, and it was going to, and so we wanted to make sure that if we were going to study it, that we were going to be able to be successful. And so we sent a glider out to the eddy edge, to look for evidence of this, and it turned out that it was there. So this is the eddy center, and we could see sloping isopycnals. This is the mode water eddy, and around the sloping isopycnals, we see these deep chlorophyll intrusions. And this is evidence of vertical advection at the eddy edge, pushing properties from the surface deep into the ocean. And this is nothing new in, this can be seen in autonomous assets in eddy-rich regions, but we wanted to be able to look at this further. And so mainly asking two questions, which is, does the biogeochemical content and community structure vary across the front? And is there a relationship between the subducted, so the subducted are the deep waters, and the upper ocean? This was a two-ship experiment, including the, that was led by Heidi Sosik on board the Ocean Twilight Zone ship, the Sarmiento, towing Heidi Sosik's shadow graph profiler, taking pictures of zooplankton and small fish, which is what we're seeing on the right-hand side. And behind was the RSS Discovery, the ship that Sasha and I were on, and we were looking for evidence of these subducted waters to go ahead and sample with a CTD. After a while of not seeing anything and biting my nails, wondering if we had missed it all, we began to see this deep chlorophyll intrusion at depth, and we knew that this was the signature that we were looking for, and it's happening right at this temperature front. This is temperature here. And armed with that information, we moved Heidi was sending us this data over WhatsApp, minus the pictures. And so we were able to plot them up on MATLAB and pinpoint exactly where the ship should be, where the Discovery should be in order to do these CTD casts. And so what we're seeing here are the CTD casts from the Discovery lining up really nicely with the shadow graph towed data from, or the towed data from Heidi's shadow graph, giving us confidence that we are indeed sampling the same waters that the shadow graph did. And you might be, if you have a keen eye and you've been looking at the pictures, you might be noticing that there's a lot more activity since the movie has gone on, and that's foreshadowing a little bit about the different distribution of things inside and outside of the eddy. All right, so this is what it looks like from a physical oceanography perspective. This is the eddy center. This is that big, huge mode water core. And this is that warm filament that's being sterned around the eddy edge. And we can see a class of isopycnals where we have enhanced chlorophyll. And it's also enhanced in percent oxygen saturation. Suggesting that this is newer water from the surface. And we want to be able to compare this with the CTD samples. And so we do a water mass analysis, a water class analysis, and we identify really what you need to know here for the main results that I'm going to show is that anything that's interesting is going to be red. So this is the subducted waters that we're seeing here. We classify as being red. If we just ask ourselves, well, how much more chlorophyll is there here? We compare the amount of, this is a ratio between the chlorophyll that is in the center of the eddy and the chlorophyll that is across the eddy at a given depth. And so we can see that while chlorophyll might change by a couple of percentage and POC measured from backscatter, and chlorophyll might change by several percentages across the eddy, within the ocean interior, this subducted region shows elevated chlorophyll of 2 to 3 times what is in the eddy center. So this indicating that this is a significant amount of chlorophyll than is in other places at this depth. All right, and we can start to look at nutrients here. I thought this was some of the cooler results that came out of this. And so what we're looking at Redfield ratios of nitrogen and silica to phosphorus, as well as nitrite. And what we're seeing here at depth is that around 300 meters, we're seeing that all of these are around their background values, their Redfield ratios. And in the, but if you look in the surface, you can see that, that things are very different. But that's also not surprising because we know this is a biologically active time and region of the ocean where we can see active phytoplankton growing, and that at this time, or at least in the recent past, some of these phytoplankton were diatoms. But what gets interesting is when you start to look below the mixed layer, and if you look at regions away from the subducted region, so inside the filament or inside the eddy, outside of the subducted regions, you see that these return back to their red field ratios very quickly. But if you're in the subducted waters, you can see that you begin to deviate from red field, indicating that these were recently in the mixed layer not that long ago, suggesting that these were in fact moved down to depths. All right, so in addition to the CTD casts, we have really interesting imaging from Heidi's shadow graph profiler, which collected 10 million regions of interest. It's really a big data problem. And they were divided up into classifications using a 29 category classifier. And we can see some of these beautiful zooplankton images that were captured by the shadow graph here. And this is all work that Heidi presented at Ocean Sciences, not this year, but 2 years before. And so these are Heidi's slides. And we could just ask, what do the distribution of these different organisms, the most abundant organisms, look like across the front? I should remind everyone where we are. This is the eddy center, and so this is the eddy edge. And you can see that in the eddy center, we have a high concentration of appendicularians, rotularians, and ctenophores, but that crustaceans tend to be dominant in this water that's being wrapped around the eddy edge. And so if we want to ask a low-order question, does the community structure change across the front, I think that an image like this is a resounding yes. We can also look at how these are distributed in the different water masses. Oh, I forgot that Heidi's, this is Heidi's cursor, not mine. So you can probably follow that because she was probably talking about something very cool. You could see that some of these were distributed across across the different water masses across the eddy, with some really interesting evidence that maybe there's some zooplankton that happen to be whose community happens to be preferable to the subducted waters, but more work is needed to look at this. I think in addition to the type of zooplankton across the region, I think it's also really interesting just to look at different particles. And that's, for those of you that do this, this is no surprise, but for me it was a surprise that the majority of what it's identifying isn't the charismatic zooplankton, but is actually different particles, orders of magnitude larger, concentration, and that's what we're looking at here, different size classes. And you can see that in these, which can be divided up into different size classes, really do follow some of the features that we're seeing. So this is the isopycnol of the subducted waters in pink, and you could see that in small size classes really can maybe be thought of as active passive tracers here being subducted along. But as you get to larger and larger size classes, you're seeing some sort of combination of both advection and sinking, similar to what Dave was showing earlier. Oops. And again, we can divide this up into distributions of water masses, showing that larger aggregates were more prominent in the eddy center and tend to have a pattern as if they're sinking out, whereas smaller sizes have this pattern of both being advected down in the subducted waters and sinking simultaneously. All right. And you know, this was just one eddy edge, but from the chlorophyll, we can see that this deeper chlorophyll signature can be seen throughout the eddy periphery. So some of the insight that we gained from the edge experiment could tell us more about processes happening around the eddy periphery and other eddies in the region. So this is the eddy center, that doming structure, and these are the eddy edges. All right, so to wrap up, I know that was a lot of information, and hopefully there were some cool movies along the way, but I just, I guess what I want you to walk away with was that there was a lot of effort, an immense amount of effort made in the sampling design and sampling execution of this field program in order to be able to achieve the goals of obtaining Lagrangian rate experiments in the eddy center, as well as being able to make observations of the lateral variability and eddy periphery. And I think that we did a really nice job, and that a lot of really cool results are starting to come out of that. So yeah, there is, in the eddy center, we see particle aggregation and sinking, and we have a nice time series of that. In the near surface, we can see lateral stirring, but and the importance of storm-driven Ekman currents at kind of broaching the boundaries of the eddy periphery and its interior. And then this example of subduction and vertical advection at the eddy edge that is carrying a lot more than just chlorophyll down to depth. With that, I'll take questions.
So what we'll do for the questions is I'll call on people and pass around the microphone so people on Zoom can hear. So do we have any questions?
Hello. That was fantastic. I love the three-dimensional movies. I was curious about how you guys actually managed to track down these cores and sample in that area, because it seems like you captured that diamond structure really well. But did it move around at all, or were you worried that you were going to miss that key part?
Yeah. So I mean, it was a multi-step process, but it was. So in all of this mess, you could see that, and it's going to show up soon. Where is it? Oh, here. Here, there's there's a little eddy. There's a little circular disc.
Oh, in the center?
Yeah. So that little circular disc that is moving is an eddy center product that a shore team on board was downloading satellite images, doing their Okobo-Weiss analysis, getting an eddy center, and shipping those coordinates or sending those coordinates to the ship every day. And every day, I would sit there and I'd redirect our whole sampling to be in the eddy center coordinates, so not Latin long coordinates. So we had this diamond structure sampling or it was a bow tie that we would try to match, but that whole bow tie was in a floating reference frame of that little disc. And then later we could use the ship ADCP to redefine another eddy center and all of the profiles, looking for that diamond structure. Like you start to combine all of those and you could say, all right, these are all lining up. Like,
That's a good sign.
yeah, that's a good sign. So that's kind of what gave us confidence. But while we were out there, it was just this satellite-derived eddy center product.
Gotcha.
And there is an eddy center product, that is made out of Dudley Chelton's products out of OSU, but those are all post-process, and so we kind of had to start from ground one and create our own eddy center product, which happened to work quite well.
I see. But the sampling here is pretty much a real-time, very involved process trying to track that.
Yeah, yeah. I mean, it's pretty cool that we were able to have the communication ability that we.
Yeah. Great. Thank you.
You're welcome.
Looks like we have a question on Zoom. And the question is, what about shipborne ADCP data through the eddy field? Did you have that data, and did it show anything
Yeah,
interesting?
we did. So we didn't have, we started adding that in real time, but again, we had all ships had ADCP data, and at least the ones on the Discovery and the Cook, we used in real time to do some eddy center products, but you only have them back in time. But they show some really nice evidence. There's also some internal wave fields and some near inertial oscillations from the storms, and so we had to average over several days, but they ended up being very useful in coming up with an eddy center product. Yeah, thanks for the question.
I was wondering, do you think that any of these physical dynamics that you've described here could be applied to other dynamic physical locations, like, for example, the California current or coastal areas, or is this really specific to the dynamics in the North Atlantic?
Yeah, that's I mean, I think the dynamics themselves are can be applied, of course. I mean, they're going to look different if you have a California current upwelling regime, but that does create that's evident. That's more clear evidence of the dynamics between Ekman dynamics and mesoscale dynamics, and where you will have really nice subduction around the edges, and where the turbulent boundary layer will kind of take an interesting path because of its interactions between the turbulence and the mesoscale currents or submesoscale currents. But how it will look will be different based on like all the small things. But I think that in general, you could say that the things that are important here are going to be important there as well.
There's another question from online from Basil. Great talk. You mentioned that a lot of particles were retained in the geostrophic velocity. Were those particles simulated or floats?
Oh, yeah. Okay. So the ones that I showed were just simulated using a poor man's particle tracking that I did on my computer. But you could see, and they were backtracked because I wanted to see where the particles came from. But I'll just show this movie again. It's turning out to be really useful. So from here, you might see all of these like little circles, the Ds. Those are surface drifters. And you'll see that very few of them actually stay inside the eddy. And they're swirling around because of these wind-driven near inertial oscillations, but they're also getting ejected out of the eddy. I can't remember if it was Dave or probably Ivona called it the conga line. So I think we're both seeing it in the simulated data that I backtrack, But there's evidence that, that this is also happening in these surface drifters. Yeah, thanks.
I'm going to call in myself here. So did you choose an anticyclonic eddy for convenience because it's retentive, or do anticyclonic eddies play a special role in carbon export?
Yeah, I think the former. So it was that the idea that the anticyclonic eddies tend to have a longer life, or that's what we imagined would happen. And you know, it turns out that like choosing 3 eddies, you'd think that like you've gone through all this work, this whole eddy field, and that you've nailed 3 eddies and that they would all be good. And it turns out only one of them was. And so I think I was a little bit surprised at that. But it was retentive. It did start to morph a little bit throughout the campaign, but it stayed retentive for the month we were there.
Another question from Basil online. Does having so many assets on the surface tamper with the velocity field?
It doesn't tamper with the velocity field. It's a good question about how much the assets you're towing or putting in the water are actually interfering with the measurements that you're trying to take. But the ADCP data that we were getting from the ships had an upper bound limit of 20, 30 meters, and deeper. So the surface assets weren't interfering with those measurements.
Well, I think that's it for the questions. So let's give Leah another hand. Thank you.
Thank you.